Okay, we'll go ahead and get started. Really excited to be introducing our speaker this month for the INN Seminar Series, Dr. Jason Chan, who's a Michael Smith Health Research Scholar and Movement Disorders Neurologist at Fraser Health, working out of the Jim Pattison Outpatient Clinic down in Surrey. Jason has kind of his CV is a list of stops of some of the best movement disorders programs in North America. So I'm going to go through it pretty quickly, but it's very impressive. Got his bachelor's at Queen's before doing an MD PhD at Western. From there, did neurology residency in Calgary, and then moved to fellowship training in movement disorders at the Fixel Institute at University of Florida, which is one of the best sort of imaging research programs for movement in the world, probably. And there he was supported by a Parkinson Foundation fellowship. Since getting to BC in probably the last year and a half, he's already set up really a world-class clinic for Parkinson's care in the Lower Mainland here, including a staff of nurses, physical and occupational therapists, speech language pathologists, pharmacists, social workers. So really this like holistic model of self-contained care within this clinic that I think is really impressive. And recently, we were just chatting about this, he also became the first neurologist in the Lower Mainland who is also working with DBS programming, so able to actually program people's deep brain stimulators that helps improve symptoms and takes care of those folks. So yeah, lately we've been working together. We're partnering on some, I think, really exciting neuroimaging research in patients with movement disorders. And I think we're really lucky to have Jason in the Lower Mainland, both as a clinician and as a researcher. So really excited to hear his talk today, and take it away, Jason.
Thank you, Alex, for the really great introduction. And I'm really happy to be here to speak to an SFU neuroscience crowd, just to get a sense of like who's in the physical audience. They can't see the Zoom. How many of you are undergrads? Grad students? Okay. Postdocs? Yeah. Faculty? And I think I saw Brianne on the Zoom beforehand. So welcome all. So today I'm hoping to give you a bit of clinical context into Parkinson's disease to try to understand a bit of the research we are doing. Then I'll give you kind of an outline as to how I've been involved in research over the years, leading to the work we're doing now in BC. So like Alex said, the work that I'm doing right now is funded by Michael Smith and Parkinson's Society of BC. No really relevant disclosures outside of that. Okay, so we're going to start by going over Parkinson's disease symptomatology with somewhat of a focus on cognitive impairment, and then we'll explore different techniques by which we can investigate Parkinson's and other disorders with a focus on field potentials, so local field potentials from intracranial recordings, and with an overlap into MEG and EEG. And then we'll talk about Parkinson's disease primarily, but also extends to other movement disorders from the perspective of network disorders and oscillopathies, so pathology that occurs when brain waves malfunction. So Parkinson's disease right now is the second most common neurodegenerative disorder after Alzheimer's disease in the world. But what makes Parkinson's quite interesting is that it is the fastest growing neurologic disorder in the world. This figure, I think, was made quite a few years ago, and it estimated that the worldwide population of people with Parkinson's would hit around 13 million by 2040. It is now 2025, and that number has surpassed 12 million already. So the rate of increase is actually faster than predicted, and it is the fastest growing neurological disorder in the world. You can see from this graph, men are more affected than women, typically at a 2-to-1 ratio, but the same holds for both sexes. That as you get older, your risk of Parkinson's is higher. So what is Parkinson's disease, right? I think we all have an image of what it may be or what it may not be. And some of us may have closer interactions with Parkinson's than others. So when we teach like medical trainees, the mnemonic that stands out is TRAP, T-R-A-P. Where the T stands for tremor, and that's a tremor in a resting state, more so than with action doing things. Rigidity or stiffness of muscles. A for akinesia or bradykinesia, so slowness of movement, but in a very specific manner, such that slowness is more obvious with repeated effortful movement. And P for postural instability, leading to falls. an imbalance. But the key point is that the bradykinesia, the slowness, is essential. So many people think that you need to shake to have Parkinson's, but that is actually not true. Many people with Parkinson's do not shake at all. So in this example, you can see a man with a resting tremor more on the left side than the right side, kind of comes out when he holds his hand up. So a key feature of Parkinson's is that typically the motor symptoms, the movement symptoms, start on one side of the body. And as it progresses, it can involve the second side, but there usually is persistent asymmetry in what they experience. In a moment, you'll see him make different hand movements. Right there, right? So you can see that right side, keep that in mind as he goes on to the left. And the left side, he has a lot harder of a time moving that hand. So that's that bradykinesia we're talking about. He has a bit of it in the right, but mostly it's on the left. Okay. The unique feature of Parkinson's is that it is what we call levodopa responsive. As we understand Parkinson's disease, the substantia nigra pars compacta degenerates. Those are the dopamine-producing cells for movement in the brain. And so by the time people become symptomatic, about 70%, about 70% of their dopaminergic neurons have already degenerated and died. But what that means, though, is the main treatment we have for Parkinson's is replacing the dopamine that their brains do not make. And virtually everyone will respond to levodopa, whether they appreciate it or not. Okay, so we focus a lot on motor symptoms in Parkinson's disease because that's the obvious feature, right? If someone can't move well, if they're shuffling as they walk, if they have a tremor, it's very easy for someone to say, like, is there something going on there? But what's important to realize about Parkinson's is that Parkinson's disease also has non-motor symptoms, and often those are more debilitating than the motor symptoms themselves. One thing that's important to keep in mind, you can see in the bottom figure there, with the green line, is that non-motor dysfunction often precedes motor impairment, so that we can't make a diagnosis until the movement symptoms come out. So in the prodromal phase of Parkinson's disease, people can lose their sense of smell. But you can see that that's not a very specific symptom, and COVID's a really good example of that, right? When everyone lost their smell, it doesn't mean that everyone's going to get Parkinson's. Something that is more specific before the movement symptoms start is what we call REM sleep behavior disorder, when patients act out their dreams at night, and there's a high conversion between that and Parkinson's. Constipation is very common as a non-movement symptom, as are things like depression, anxiety, and other psychiatric manifestations. As the disease progresses, all of these symptoms can get worse, can become more prevalent and persistent over time. And like I mentioned, it's that continued progression of both the motor and non-motor that all contribute to disability. One of the non-motor symptoms that we're going to focus on today, and one that I'm very interested in, is cognitive impairment. And so cognitive impairment increases with age in Parkinson's disease. Roughly a third of patients overall will have some form of mild cognitive impairment. So that's symptomatic impairment that they experience, but that does not necessarily get in the way of their day-to-day function. Once it does get into the way of their day-to-day function, we call that clinically dementia. And Parkinson's is a cause of dementia, just like Alzheimer's is, and just as many other conditions could be. Right, so then these symptoms, right, are monitored in the clinical sense using screening tools. As you can see here, more so for the clinical audience, it's the latter three that we rely on, more so in terms of their specificity for things like executive function dysfunction and visual spatial function dysfunction. So when we talk about clinical cognitive function, what we're talking about usually are the clinical cognitive domains. And these are the ones that neurologists and psychiatrists and psychologists are quite familiar with. So they are attention and working memory, executive function, longer term memory, language and visual spatial function. And in Parkinson's disease, it is the attention, executive function, and visual spatial function that are preferentially affected more so than the others. So when you talk to people with Parkinson's and ask, well, like, what is happening cognitively? They'll often describe things like difficulty focusing on conversation. difficulty multitasking and doing many things at the same time, even though they may have been able to do that years ago. They'll describe difficulty performing multi-step tasks, often because they'll forget a step as they go through them. They'll describe difficulty remembering things that just happened, and then they'll recall it, usually moments later. They'll describe difficulty with reaction time and just overall processing speed. And very commonly, they'll find difficulty finding the right word to use in conversation, which is quite impactful when they're trying to be social. That's kind of the clinical constructs of how we think about cognition and how a patient may think about cognition. But scientifically, that's only one way of thinking about it. And for me, I like to think about cognition a bit more broadly, to say that cognition involves essentially all the processing that happens between stimuli going into the brain and an output coming out of the brain, and that output typically is movement. What makes it quite natural that Parkinson's disease is a good disease in a sense to study this in, because it has all the elements of basically the whole processing pathway from sensory input to processing to motor output. And the motor output is often would be considered normal. And so in the scientific sense, you can further break these cognitive functions down into the following that you see on the screen. And for many of you who are in neuroscience or deal with systems neuroscience, cognitive neuroscience, these terms would be quite familiar to you. One thing to realize in the clinical sphere is that people can have difficulty in these cognitive functions, but not be aware of it, right? So it may not be enough to cross a threshold for them to notice in their day-to-day lives. And one really good example of that in Parkinson's disease is a phenomenon called freezing of gait. And so freezing of gait is a phenomenon where when people go to walk, they know they want to walk. They know they want to create forward progression, put one foot in front of the other, but they just can't. They'll often describe a disconnect between their brain and their legs or their body. And as much as it is a motor phenomenon, it is a cognitive phenomenon. So this is a man, I think, he was in his 70s. 8 years of Parkinson's disease, and you can see here he's trying to walk, but you can see how effortful each step is, and when he finally breaks free from the ground to make a step, those steps are quite small. In a moment here, he's trying to turn, he's going to try to turn towards the right, and you see that left foot just gets stuck there, right? So outwardly, it is very much a motor phenomenon, but this is a really good example of the interaction between impaired cognition, not necessarily symptomatically, right, but impaired brain processing and the ability to actually move. So just to summarize, cognitive impairment in PD, as are many other non-motor symptoms, are quite common. Cognitive impairment typically affects attention and executive function and can often interact with motor abilities. Contributes quite a bit to disability, quality of life, and caregiver burden. Because the symptoms that affect people with Parkinson's don't just affect them, they affect everyone around them. And the key with this is that it is often quite difficult to treat. Right? Motor symptoms, we have levodopa, cognitive impairment, much harder, as are many of the other symptoms. Which brings us to some important questions. Right? What is actually going on in the brain? Right? What could the neurophysiologic mechanisms be that underlie motor function and dysfunction, cognitive function and dysfunction? And if we can understand these mechanisms, can we then use our understanding to refine current therapeutics or develop new therapeutics to treat these symptoms? And that brings us to how do we investigate some of these phenomena? So many of you are probably familiar with this slide. This shows a variety of neuroscientific techniques across different scales of temporal resolution and spatial resolution. These techniques can be used in either humans or animals, depending on how invasive they are typically. And you really need a complement of all of these to understand the brain fully, from kind of the microsecond scale to the second scale, from the micrometer scale spatially to the whole brain centimeter scale. And so these are kind of what we focus on. And what we'll be talking about today relates to field potentials that we can record cranially or extracranially from the brain, which also relates to EEG and more specifically for the group here at SFU, MEG. So let's start by going through intracranial electrophysiology. So this is typically done by inserting a microelectrode into the brain itself. This can be done cortically or subcortically into deep brain structures. And the microelectrode typically picks up potential changes in the immediate vicinity of the electrode tip. And so typically is used to pick up action potentials or spiking activity of neurons. You can see here that microelectrode is right next to blue and red as the neurons, and so they pick up the blue and red signal. And when we're talking about local field potentials, what we're really talking about are the synchronized synaptic activity of a group of neurons. So now instead of recording a single neuron or two, we're really recording from thousands at a time. And it's the signal that can help us better understand how different brain areas could interact with each other. So one way of doing that is you could look at the raw signal, right, which is shown as an example there. But this raw signal can be further decomposed into its frequency components using time frequency analyses, so techniques like the Fourier, Hilbert, or wavelet transforms. So decompose. And then based on the peaks in the power spectrum, these frequencies can then be further subdivided into different frequency bands. And so these are the typical bands that we talk about. So delta being 0 to 4 hertz, theta being kind of 4 to 8 hertz, alpha somewhere between 8 to 12 or 13 hertz, beta between that 12 to 13 to 30, and then anything above 30 being gamma, and then you can talk about like high frequency oscillations, that sort of thing. But you can basically break them down to different categories, which then may reflect different functions in the brain. So one thing that there's been more and more evidence for is that oscillations help different neural groups and brain areas communicate. And so here you see a schematic of 3 different brain regions, red, green, and black. And you can see that if 2 areas, being red and green, are synchronized in terms of their oscillatory activity, that facilitates neural communication, that facilitates the passage of information via action potentials. And by extension, then, if you look at whole brain networks, oscillations can then be used to facilitate a certain brain area's participation in multiple different networks. Right, and so you can see kind of process 1, process 2, process 3 represented by different configurations of brain area interactions, and it's thought that oscillations can help the brain do so. So going through the individual frequency bands and their functions, theta activity is well studied in the hippocampus, and so it's been well implicated in memory, specifically memory encoding and retrieval, and as well as working memory. There's also some role for it in monitoring of responses and monitoring of outcomes. And with it being a slower oscillation, an oscillation you can actually count if you were to count it, that slow nature has been thought to be ideal for creating windows of association between different brain areas. This is just an example of a study that looked at theta activity. You can see that midline theta, which is increased across all these conditions, is associated with novelty, conflict, response monitoring. The alpha band has been typically thought to be associated with inhibition, or in other words, modulation of excitability. So here we see an increase in alpha signal actually suppress gamma activity in the brain, and it is also thought to be used by different networks in different manners to subserve their own functions. So on the top, we have the cinguloopercular network, which is thought to be involved in creating alertness, and it may use alpha in such a way that it uses it to suppress whole brain activity to maintain that alertness. The middle shows the dorsal attention network, and that's typically thought to be involved in selective attention. So it may do so by focally suppressing alpha to allow that image or that input or what have you to kind of break through. And then the frontal parietal network, involved in executive function and adaptive control, may shift alpha in different ways to basically configure different brain areas to serve its function. Beta activity is a huge topic in Parkinson's disease for the reason that it has been shown quite well to be involved in maintenance of sensory motor states as well as cognitive states. The inability of someone to move, or when we're not moving, we're holding still, typically is associated with increased beta. And in Parkinson's disease, we typically see increased beta above what is considered normal, and that has been well associated with their slowness of movement and with their stiffness. So here on the left is an example of a paradigm where a patient is asked to move. Right, the W there is the warning signal, the G is the go signal, and M is when they make that movement. And you can see, like, right up until they make their movement, their beta starts to slowly decrease. And that beta decrease is what we think releases that movement and allows it to occur. And is more obvious when people are off levodopa untreated. Like I said, beta is also involved in maintaining cognitive states. And so this is an example of an experiment in a macaque monkey where they were asked to remember one object, wait for the second object, then decide which object they had just seen. And you can see on the bottom there that there's increased beta during that delay period when they're having to hold that object in their mind or in their working memory. So that kind of brings me to the work that I did when I was in grad school. And at that part of my career, I was very interested in how oscillatory activity and interactions between different brain areas contributed to cognitive control. And so just to kind of give an overview of brain areas that are involved in cognitive control, typically we think about these ones that are shown here. I'm not going to read them all, but you can see them right there. And these are the brain areas that typically are activated no matter what task you do. Right, and so for that reason, there have been some, including John Duncan, who've called these the multiple demand system. They are areas of the brain that pop up time and time again for different tasks, different cognitive functions. And so when I was thinking about cognitive control, the area that interested me most was the prefrontal cortex. And of the prefrontal cortex, the two areas that come up time and time again are the ones shown here, the dorsolateral prefrontal cortex, DLPFC, and the dorsal anterior cingulate cortex, or actually the entire anterior cingulate cortex, the ACC. And so to investigate the function of these brain areas in cognitive control, we looked at a way to more reliably link these brain areas to behavior in a causative way. And to do that, we turn to cortical cooling. So what we see here is a cryo loop, and these cryo loops can be implanted onto the sulci or into the gyri of brains. And by passing chilled methanol through the input and the output, essentially through the loop, we could actually bring brain temperature down to below 20 degrees Celsius, and that's the temperature at which brain activity, active potentials turn off. So we use these cryo loops in macaques to basically cause reversible lesions and to see what those lesions did to brain function and to behavior. And the neat thing, of course, with this is that it's quickly reversible, so each animal could be used as their own control. So in the first experiment that I'll talk about, we were curious about the relationship between the DLPFC, which is that prefrontal area, and the superior colliculus, or SC, which is an oculomotor output nucleus. And we did this, so we cooled the DLPFC bilaterally with these cryo loops while recording from the SC, while these macaques performed an interleaved prosaccade and antisaccade task. If you're not familiar with what these tasks are, the prosaccade is essentially where you ask someone or an animal to look towards an object that pops up. And that's a very easy response to make because we very often will look towards a novel stimulus. The antisaccade task is the exact opposite, right? So now we're asking someone or an animal to look away from the object we present. And this is more difficult in the sense that you have to inhibit a very much a reflexive or a habitual response to look towards something, and then generate a saccade in the complete opposite direction into empty space. Right, so you can see that one is easier, one is harder. And so when we looked at the data here, we found that bilateral DLPFC deactivation reduced beta activity in the preparatory period. So in the period leading up to when the animals were about to make their move, either with a prosaccade or an antisaccade. And we showed this with both task conditions. And one interpretation here is that the DLPFC via beta activity mediates communication between the two structures and allows for that task signal to kind of come through and allows the animal to then make the correct move. In another study, we looked at the relationship between the two brain areas I had mentioned, the DLPFC and the dorsal ACC. And similarly, we implanted the cryo loops into the DACC while recording from the DLPFC, again using prosaccades and antisaccades, but this time in block configuration. And we found that bilateral DACC deactivation reduces alpha activity for prosaccades in that preparatory period, but in contrast with antisaccades, did not affect alpha activity, but actually reduced theta and beta. And this may relate to the differences between the two task conditions. The antisaccades are more cognitive demanding, and so perhaps that's where theta and beta come in, is to help the brain and help the animal behave in a more cognitively demanding way. Furthermore, we found that beta activity distinguishes correct trials from error trials. And so typically on correct trials, that beta was higher. On error trials, it was lower. And you can use that as a marker of, did I do a good job or not? Or did the monkey do a good job or not? And what we found is in cooling the DACC, beta in the DLPFC was reduced. So the difference between the beta in the correct trials and error trials was reduced. And so perhaps beta is actually needed to help discriminate what that response was. So to summarize this, we can use reversible deactivation to help establish causal relationships. Of course, there's still an element of correlation here, but it is more convincing if you can shut off something and then show that something happened. The studies that I showed you suggest that beta oscillations help mediate top-down control. And then in terms of DLPFC, DACC, oscillations help facilitate the performance of cognitive demanding tasks, being the antisaccade that we used. At the time, this was done actually about 10 years ago, it added to the growing body of evidence supporting oscillator activity in cognition. And really in the last 10, 15 years, that's been a really big topic in systems neuroscience. One thing to keep in mind here is that many studies of oscillations, whether they're related to cognition or movement, are primarily studied using two different groups, right? So either animal models for the invasive studies or non-invasive studies in humans, often involving MEG, EEG. And you can see, though, that doesn't, so the invasive studies in animals don't necessarily represent humans, especially humans with disease conditions like Parkinson's. And so we're going to move on and then talk about Parkinson's and how we actually study these in disorders and in humans. So it's hard to talk about Parkinson's without bringing up the basal ganglia. Right? So for many of you who have studied neuroscience or have an understanding of Parkinson's, that you probably will have seen some schematic of the basal ganglia. And we know from a variety of different study types, right? So lesion studies, functional imaging studies, neurophysiological, electrophysiological studies, as well as Parkinson's disease itself, that the basal ganglia are critical to motor control, as well as cognitive control. And beyond the basal ganglia, they participate in cortical basal ganglia thalamocortical circuit. So really the whole brain, right? To basically allow people to move and think in an appropriate manner, and it's dysfunction in these circuits that then cause symptoms to arise. When we talk about the basal ganglia, we can talk about different parts of it. The two structures I'll focus on today are the subthalamic nucleus, or STN, and the globus pallidus internus, or GPI. Both of these structures, although well implicated in motor control, also have different subregions that contribute to cognitive function and affective function, so mood, anxiety. And you can see here, this is a schematic of the STN using tracers in animals. And typically, the dorsal STN is thought to be the motor subnucleus. The ventral STN is thought to be the cognitive subnucleus. Similarly, when you look at the GPI, you can see that split as well. So the motor being more posterior, the cognitive part of it being more anterior. And this pattern kind of holds true throughout the motor system or throughout the basal ganglia, the thalamocortical system in the brain. And the question becomes, well, in humans, how do we study this? And uniquely for Parkinson's disease, or not entirely uniquely, but a unique aspect of Parkinson's disease is that one of the treatments involves intracranial study. And that brings us to deep brain stimulation. So deep brain stimulation is a therapy that's been used for well over 20 years in Parkinson's disease to help control motor symptoms, typically bradykinesia, rigidity, tremor. Can also be used to control dyskinesias and dystonias in Parkinson's. And since the early days of DBS, right, stretching way back into the 70s, and even before that, recording electrodes have been used to investigate electrophysiology. And so those microelectrodes we talked about are often used in DBS surgeries to help the surgical team as well as the neurological team identify where they are in the brain. And often this can be done with concurrent recordings to help better understand physiology in Parkinson's. Now, much of the focus has been on motor control in Parkinson's, but there is some cognitive literature as well. Okay. These are just an example, a slide of examples of actual DBS electrodes that are typically implanted. And now in 2025, we can actually use some of these electrodes to record brain activity in real time in patients who have been implanted with these leads. So much of basal ganglia circuitry was kind of sorted out using single unit studies in both humans and animals. And this is the typical diagram of the basal ganglia that we see in textbooks and presentations and lectures. So we talked about LFPs and motor control in Parkinson's. We saw that figure earlier, right, where reduction in beta is required to initiate a movement. Beta activity in the STN is well studied and is typically, it's that peak in beta activity that's thought to be related to the disease. You can see in that figure on the top there that if someone takes their medication, that beta peak decreases. And that beta peak decreasing, the beta power decreasing, has been associated with improvement in motor function, measured here with the Unified Parkinson's Disease Rating Scale, or UPDRS, where essentially lower beta is, usually a bigger increase in beta is associated with a bigger improvement in their motor symptoms. Similarly, when we look at the GPI, we see that as well, that there is increased beta activity in the GPI. Similarly, if you give someone with Parkinson's medication, that beta peak in the GPI also decreases. The decreases for both GPI and STN are also seen with DBS, right? So if you turn the DBS on in these patients, that beta signal decreases as well. And again, associated improvement in bradykinesia and rigidity, not so much the tremor. We see this beyond the nuclei as well. So when you look at coherence between different brain areas, so GPI and primary motor cortex, STN, primary motor cortex, we see that the coherent signal is also associated with symptoms, with higher coherence being more symptoms, less coherent being improved symptoms. Although I said that most of the studies are done in motor control for Parkinson's and LFPs and basal ganglia, there are some studies looking at cognition as well, but typically very much focus on task performance, right? So they'd have patients in the OR or after they are immediately implanted, run through a bunch of cognitive tasks, and then try to associate changes in electrophysiologic activity with different phases of the task. So here we have someone who's performing, or a study where they had people with Parkinson's perform a Stroop task, right? So typically reading the actual color of the of the stimulus rather than the written color. And you can see here in the figures that on the left side, the dotted lines represent reaction time. And what's kind of key here is that data activity in that bottom left there is increased when people are presented with incongruent trials. So this idea that data may be involved in kind of identifying or processing the incongruence and the conflict that's present there. In another example, they had patients perform the flanker task, where so again looking at incongruent stimuli, and they showed that increased activity was present again when those incongruent stimuli were presented. And what was interesting here in this study was that increased data led to faster reaction times, right? So more so linking it to behavior. In another study, they looked at basically within trial conflict and response inhibition, and what they showed between the medial prefrontal cortex and the STN is that there is increased coherence on those trials with conflict. So when they're asked to not go versus go. And what was interesting in this study then, because we've talked mostly about theta so far, right, in terms of the STN and cognition, is that beta does play a role, but it doesn't seem to play as much of a role in preparation and execution of the response. It seems to be more monitory. So what we're seeing here is that beta, we see an increase in the power on the subsequent trial if patients were to make an error. So not in the current trial, but the subsequent trial, right? So not as much of an immediate impact on the task, but perhaps a monitory corrective response after the fact. Okay, so far we've talked really about oscillatory activity during task performance, but how cognitive impairment or cognitive abilities in people with Parkinson's is reflected by peer oscillatory activity at rest has been relatively unknown. There's been some studies that have looked at this more cortically, right? So the first one that I'll highlight is this paper here that used MEG to look at Parkinson's patients with either normal cognition, mild cognitive impairment, or full-life dementia, and they found that the MCI group had increased whole brain resting theta and beta activity, which, again, perhaps supports the role of those two in cognitive performance. And the next study by Alex Wiesman, right, in MEG and PD patients found that parietal slowing was associated with cognitive impairment and motor impairment, and that perhaps frontal slowing was associated with compensation in their function. And like I said, these studies, being MEG, right, non-invasive techniques in humans, are very cortical. The question becomes, well, is there a neurocorrelate of cognitive impairment in the basal ganglia? And these are the ones that we could actually manipulate and treat using techniques like DBS. So while I was in Florida, I was part of a study that looked at this using intracranial DBS recordings. So the data that I'm going to show you is from patients who underwent either unilateral STN or GPI implantation for their Parkinson's. And we kept it unilateral to avoid a confounding effect of the second lead. So these patients came into their OR as they typically do, and is standard practice off their medications, so in the off state. And after the lead position was confirmed using microelectrode recording and lead side effect and benefit testing, we recorded a minute of LFP activity at rest. All of these patients, as part of standard protocol, underwent comprehensive psychological testing before their implants, typically anywhere between 3 to 6 months before we allowed it to be up to a year to be included in the study. And we came to a total of 89 leads across 89 different patients, so unique patients. This is the breakdown of the leads. So it is weighted a bit more towards GPI than STN. You can see the age breakdown as well as the sex breakdown. Typically, GPI patients are a bit older, and that relates more so to why we implant GPI over STN. Typically, more men than women, as is typical of the demographics of Parkinson's. And their mean UPDRS, so their motor function, typically was in the 30s. This is a schematic here. Oh, so yeah, and the analysis then used the two middle contacts of the PDS leads, typically because those are the ones that the surgeons would put right in the middle of the nucleus you want to treat. Keep in mind that the targeting then, right, for the nuclei is actually not cognitive, even though we're looking at cognitive function. Treatment requires putting these leads into the sensory motor nucleus, which for the STN is right next to the cognitive subnucleus, whereas in the GPI, there can be more space in between, which is important later. So this is a schematic of just power spectral densities, an example from STN and GPI. You can see both of these have clear beta peaks, right, which we expect to see in Parkinson's disease, right, 22 hertz for the STN, 24.5 hertz for the GPI. So in terms of the results, right, these patients did a battery of neuropsychological testing. And here what we're seeing is that there is a relationship between digit span or DS and the trails making test A and B, which are shown here. So for those of you who are not as familiar, digit span requires people to repeat a series of numbers, either forwards or backwards, and you're scored based on how well you do in terms of how well you basically match the numbers. In the trails test, A is essentially connecting dots in ascending order for a single number series. The trails making test B is a bit more difficult in that you're then alternating between letter and number. So what we're seeing here is that in that second from the left plot, increased theta activity is associated with a lower digit span score, so worse performance. Trails A and trails B, a higher theta power, resting power, was associated with a higher time on both tests, so again, a worse score. And these three tests are tests of attention and executive function, which we had mentioned before are preferentially affected in PD. So it's quite interesting that these were the tests that were affected. When you look at other tests like the Stroop or the Hopkins Verbal Learning Task, which is a memory task, Boston Naming Test, which is a language test, and the JOLO or Judgment of Line Orientation Test, which is a visual spatial test, these really didn't show that same correlation, and it's really those tests of attention and executive function that showed that theta relationship. Interestingly, we did not find a significant signal in the GPI. Now, that doesn't mean that GPI is not involved in cognition, because we know it is, but it may just relate to the techniques we use and where we're recording from, the size of the GPI, right? So, and the other thing is mean resting power may not be the best measure, right? Because there are other oscillatory parameters that we did not talk about in these slides, with like peak power, beta bursting, all that sort of stuff that could still be important, but was not looked at as part of the study. And so that's to say that the basic entity participates in a widespread circuit that is involved in cognitive function. To summarize, theta power seems to be associated with performance in attention and executive function, which we know are preferentially affected in Parkinson's, which leads us to think that, could theta be a biomarker of cognitive performance, right? Both in the resting state, as we saw before in previous studies, during actual task performance itself. And then because the STN and GPI had different responses, it is possible entirely, right, that they have different mechanisms of involvement in cognitive function. So one thing that's kind of interesting about all the studies we've talked about so far in PD is that they typically are done in a single state, at a single point in time, right? So these are patients who are either off their medications or on their medications, and we take a snapshot of their brain activity or task performance or motor performance all at one time. But the reality of Parkinson's disease is not that, right? And any person with Parkinson's disease, if they've had it long enough, will tell you that it fluctuates, and it fluctuates a lot, right? So what I'm talking about here is that symptoms can fluctuate, medication benefit can fluctuate, medication side effects can fluctuate. And also talking about symptoms broadly, so not just motor, but also non-motor. And then the other key point is that no one with Parkinson's, if you compare one person to the next, is alike, right? There's so much variability between one person to the next. And so to capture Parkinson's disease kind of in real life, we also have to capture the intrinsic variability of Parkinson's, either internal to the person themselves or when you compare from person to person. And that kind of brings us to the work that we've been getting up and going here at BC. So we recognize Alex Wiesman. So, but, I think I'm very fortunate to have started around the same time in BC as Alex. And I remember we met very early on. I think it was like summer 2024, right? And very quickly, we kind of hashed out 3 questions around the variability of symptoms and medication effects in Parkinson's. So number 1, right? How do brain networks and neurophysiology change over time? And we're not talking about over time in terms of days or months. I'm talking about you take the medication typically takes about 30 minutes to an hour to take effect, variable between each person, reaches peak effect 1 to 2 hours, and then starts to wear off, right? How does the brain actually change in response to a single dose of dopamine replacement therapy, typically levodopa? The second question then was, if we can create dynamic brain activity response profiles, can we then look at how do we separate these to on-target dopaminergic effects, the effects that cause motor benefit, from the off-target brain effects of dopamine that could then cause side effects. Right? So common side effects, typically of levodopa, would be things like nausea, which is levodopa effect on the GI system, or the stasis, which is levodopa effect on the autonomic system, drowsiness, which is a cognitive side effect, dyskinesias, which are extra involuntary movements, which are definitely off-target brain effects of dopamine. Right? So how could we actually separate these two? And most importantly is the third point, right? Is how can we relate these response profiles then to actual clinical function? Motor function and cognitive function being the 2 primary areas that we were interested in, but we'll likely look at different aspects too, such as blood flow. But this is really the important one, because if we can actually understand why patients experience the symptoms they do and how their symptoms respond to medications, we can then hope to refine the way we treat them. And so this builds on work that Alex did very recently, looking at how can you separate on and off-target effects using neurochemical atlas mapping. So in this study, which you may have heard about a month ago when Alex gave his talk, right, looked at MEG in the off and on state 1 hour after patients took their medications. It found that beta activity in general was quite variable between patients. And the interesting point, right, shown really by the bottom 2 figures, is that increased beta signaling in dopamine-rich areas, especially in the inferior frontal and temporal cortices, seem to be associated with less of a motor response to their medications. Right, so this kind of sets the groundwork for the bigger study that we're planning on doing, and to get the dynamic nature of how does levodopa affect brain activity over time, plan on serially investigating patients pretty much right after the time they take their medication. So the idea is we're going to bring Parkinson's patients in, or people with Parkinson's into ImageTech to get a good off-medication baseline. So across studies, this is typically accepted to be 12 hours off medication. So the idea is you have patients take their last dose the night before, and they come into the session without any medication the day of. The idea here is to really do a good screen, get a good motor baseline on them with the MDS-UPDRS3, which has been typically the standard for measuring motor symptoms in PD, although not perfect. We also plan to look at measures of cognition, which can then be done pre and post. So essentially when they're off levodopa and when they're fully on levodopa. But the key part of the study is really to look at measures of brain activity, motor function, cognitive function over time. And so this time, I forgot to write this down, this is going to be time essentially over a 2-hour period, right, which effectively captures levodopa onset, levodopa peak, and in some people may actually capture levodopa wearing off. right? So we plan on doing blocks, 15-minute blocks of alternating MEG and clinical testing to really capture how brain activity, motor function, cognitive function change over time. And we're really excited to say that this will be hopefully be up and running by the end of the year. So unfortunately, no results to show as of right now, but I think this will be a really comprehensive data set for understanding Parkinson's, not just from the medication point of view, but from a patient variability point of view. So it'll open up many ways to answer different questions. So in terms of the next steps related to the dynamic tracking of dopamine therapy study, we're really looking at identifying mechanisms of symptom fluctuation, variability, and interaction between symptoms. As we mentioned, symptoms like motor symptoms, cognitive symptoms, autonomic symptoms, they often affect each other as well. We're hoping to identify biomarkers, right, electrophysiological biomarkers that can then help refine therapies. First off, the medications, right? Can we actually identify where a person's medication cycle they are when we do a scan? And then also help guide development, right? Either in terms of pharmacology, right? Drugs affecting certain brain areas, but also in terms of neuromodulation, right? Can we then use invasive techniques like DBS or non-invasive techniques like TMS to alter brain activity in a beneficial manner? And these same techniques, although we've been talking about Parkinson's for the entire talk, can also be applied to other movement disorders, right? Many of these other movement disorders are less common than Parkinson's, but because of that are often less studied, less well understood, and are much more challenging to treat. So whereas Parkinson's disease proper, the typical Parkinsonism, if you will, has many treatments available to it, the atypical Parkinsons typically do not. And it will be important to better understand what is actually happening in these people's brains to help better develop ways to treat them. Tremor is common enough, but can still be studied using similar techniques. Dystonia, and of course, therapeutic techniques like DBS can also be studied with what we talked about. Wearables is a hot and growing topic in movement disorders. From 2 points of view, there's increasing need to better characterize movement. Like I said earlier, much of Parkinson's disease research is based on the UPDRS scales, which is really, in terms of the motor exam, a full scale. So you can kind of see how that's not really great at discriminating fine motor control. And so that's where wearables can come in. There's also a line of thinking is, could we use wearables for therapeutics? Right? Can we use it to help treat certain symptoms, perhaps like the freezing of gait that we saw at the beginning, by having something that a person wears trigger them into making a correct response or a corrective movement. Which really then brings us to the here and now, right? So here I'm giving this talk at for the Institute of Neuroscience and Neurotechnology, and all of these things that we're doing, right, it's built on collaboration, and so it's really collaboration with the INN, with SFU, with ImageTech, and with anyone who may be interested in Parkinson's disease research, neurological research. And ultimately, the goal is, how can we improve the quality of lives of patients who are affected by these diseases, the families who are affected by these diseases, and how do we give back to the community that we all live in? And so with that, I'm hoping that we can then open up discussion and basically move this forward. To end, I just want to acknowledge the team that was kind of behind the DBS Basal Ganglia study, as well as ongoing collaborators. Two names you'll recognize, Alex Wiesman and Randy McIntosh. I don't think I'd be here if the team of INN members, including Randy, Brianne McLean, Helen Sperry, Kelly Shen, did not convince me that was a good idea to move to BC and set up my clinical practice here. So I'm very thankful for them for allowing me this opportunity. These are the funders that have been involved in many of the projects we've talked about today.
Thanks again so much, Jason.